Hi, welcome to my YouTube channel. This is Soumya. This video is on solving the KSET license question paper from 2014. The paper which I am solving is paper 3 and this is the part 3 video on solving this KSET license question paper. So now let's solve together. I'm going to start from because I have solved up to 14 questions in the part 2 video. Now I will begin with the 15th one. And 15th question is like this. The primary regulator of Leydig cell secretion is. Options are like this. Option A, FSH, that is follicle stimulating hormone. Option B, luteinizing hormone. Option C, FSH releasing factor. Option D, inhibin. First we will know what are Leydig cells. The Leydig cell have another name. These are also called as interstitial cells. These Leydig cells or interstitial cells are present near to the and this Leydig cell is going to produce the testosterone by associating with the luteinizing hormone. That's how it is going to function. And Leydig cell and luteinizing hormone both together will produce us the testosterone hormone. And coming to the options, option A, follicle stimulating hormone. This follicle stimulating hormone, it is also a gonadotropin hormone produces from the anterior part of the pituitary, and this hormone in case of females it helps in the regulating the menstrual cycle, and then it helps in the ovulation, and then it looks up to the development or the sexual development, and means it is helps to the proper functioning of the reproductive system. And this follicle stimulating hormone in case of men, it helps in the sperm production. And coming to the luteinizing hormone is also a gonadotropin which is produced from the anterior part of the pituitary, and this luteinizing hormone is going to work with the Leydig cell, as I just now mentioned, or interstitial cells, okay, to produce the testosterone. And third option, FSH releasing factor, the one which is going to help to release the follicle stimulating hormone. So that factor is called as releasing factor. Okay, and coming to the option fourth, that is option D, inhibin. This inhibin, actually as in the name indicates, you can guess it out. Inhibin means inhibit anta. It is going to stop that production. Means this inhibin hormone is going to inhibit the function. What are all the function? It is going to inhibit. In case of both men and women, this inhibin is going to regulate the production of the FSH and estrogen. Now we can able to solve this question. So the primary regulator of Leydig cell or interstitial cell is option B, that is luteinizing hormone. Let's move on to the next question. During oxidative phosphorylation, mitochondrial synthesis of ATP occurs due to option A, oxidation of glucose by glycolysis, option B, electrochemical proton gradient, option C, oxidation of NADH to NAD plus, and option D, oxidation to pyruvate to acetyl CoA. We should learn the concept of oxidative phosphorylation. Okay. And this process is actually going to start with the proton gradient. Okay, so hagagi the option B is the correct answer for this. That is electrochemical proton gradient. Oxidative phosphorylation is refers to the synthesis of ATP by ADP and inorganic phosphate by that is going to driven by the energy release from the oxidation of reductive powers NADH and FADH by transfer of their electrons through the electrons. See, as you can see in mitochondria, this process, the oxidative phosphorylation is going to take place by the proton gradients. Means here you can see the electrons. Okay, that actually helps in the moving of the hydrogen ions from in and out, from matrix to intermembrane space. Okay, or from intermembrane space to matrix. Okay, that's how the oxidative phosphorylation is going to take place. It's about the gradient through electron transport chain to oxygen. Copper is associated with which of the following mitochondrial enzymes? Option A, cytochrome oxidase, option B, succinate dehydrogenase, option C, catalase, option D, acid phosphatase. As you can see in the electron transport chain, this is complex 1, complex 2, complex 3 and complex 4. In this, the complex 1, NADH oxidoreductase, and complex 2 is succinate dehydrogenase, and complex 3 is cytochrome BC1 complex, and complex 4 is cytochrome C oxidase. So ಇಲ್ಲಿ question ಅಲ್ಲಿ ಕೇಳಿರೋದು cytochrome C oxidase. Okay, this is the one which is going to work with the copper so that The copper is going to help in stabilizing and regulating the activity of this cytochrome C oxidase. This is the complex 4, and the copper binds to the COX-1 and COX-2, the core subunits of the cytochrome C oxidase. Coming to the second one, succinate dehydrogenase. Succinate dehydrogenase, as you can see in the diagram, this is the complex 2, and it is not going to work with the copper here. And this succinate dehydrogenase, it is the only the enzyme that is it is a membrane bound enzyme of the citric acid cycle that interacts directly with the respiratory chain. And coming to the next one, catalase. Catalase, it is an antioxidant. Means it prevents the cell oxidative damage. It is going to prevent the cell oxidative damage by converting hydrogen peroxide into a oxygen and water. Prevents the cells from toxic effects of the hydrogen peroxide. And coming to the option D, acid phosphatase. Acid phosphatase, it is an hydrolyze enzyme, and it catalyzes the hydrolysis of chemical bonds. So hagagi the correct answer for this is option A. Option A is the correct answer, that is cytochrome oxidase. Moving on to the next question, 18th one, enzymes, vitamins and hormones are common in option A, being proteinaceous, oxidized in the body of the organism. Option C, enhancing oxidation metabolism. Option D, regulating metabolism. So the correct answer for this is option D, that is regulating metabolism. So these enzymes, vitamins and hormones are common in regulating the metabolism. So that is the more appropriate answer for of this question. Okay. Next, moving on to the next question. 19th one, BRCA1 and BRCA2 are involved in diagnosis of option A, breast cancer, option B, myeloma, option C, carcinoma, option D, teratoma. So breast cancer ಬಗ್ಗೆ ಕೇಳ್ತಿರೋದು, and these BRCA1 and BRCA2 are nothing but these are the ones which codes in these are the genes which avoids the breast cancer. If there is any mutations in these BRCA1 and BRCA2, then it leads to a breast cancer. Okay, BRCA1 and BRCA2, these are a suppressor genes, means these are going to suppress the cancer development agadna avoid ಮಾಡ್ತವೆ. But if there is any mutant in BRCA1 and BRCA2, this will lead to a breast cancer. And this BRCA1 is present in the chromosome 17 and BRCA2 is present in chromosome 13. And this BRCA1 and BRCA2, this might be hereditary. They may carry from the parents. Okay. So ಇದಕ್ಕೆ correct answer ಹೇಳೋದಾದ್ರೆ, the correct answer for this is option A, that will actually lead to a breast cancer. The full form of BRCA1 and BRCA2 is breast cancer gene 1 and breast cancer gene 2. Moving on to the 28th question. 28th question is like this: carcinomas are tumors of option A, hematopoietic system, option B, lymph nodes, option C, epithelial cells, option D, connective tissue. So carcinomas are tumors of, let us look at the option A, hematopoietic system. Andre hematopoietic system means it is a process of developing the blood cells, different types of blood cells ಇದೆ ಅಲ್ವಾ. So ಅದು ಡೆವಲಪ್ಮೆಂಟ್ ಆಗೋದು hematopoietic system ಇಂದ. So it is nothing to do with the tumor. Okay. And coming to the lymph node, obviously lymph node is not the answer. Lymph nodes are small cluster of cells. Okay, and they fight against the invaders. It is associated with the immune system. Okay, and these are going to fight against the foreign particles. Okay, which helps in the fight against the foreign particles. And coming to the epithelial cells, is actually carcinomas are tumors of epithelial cells. So option C is the correct answer. So epithelial cell is the correct answer. And connective tissue ನಲ್ಲಿ tumors formation ಆಗೋದು sarcoma ಅಂತ ಹೇಳ್ತೀವಿ. So ಹಾಗಾಗಿ option D is not the correct answer. So option C is the correct answer. Next, moving on to the 21st question. Replica plating is used to detect option A, mutant colony, option B, aerobic bacteria, option C, blue and white colony, option D, normal cells. First we will know what is replica plating. What is replica plating? Replica means, so ಒಂದು ಆಬ್ಜೆಕ್ಟ್ ಇರುತ್ತೆ, ಸೊ ಅದ್ರ ಎಕ್ಸಾಕ್ಟ್ ಆಗಿ ಕಾಪಿನ ಪ್ರಿಪೇರ್ ಮಾಡೋದು, ಇದ್ರ ರೆಪ್ಲಿಕಾ ಇದೆ ನೋಡಿ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ದಟ್ ಈಸ್ ದ ಮೀನಿಂಗ್ ಆಫ್ ದಟ್. ಸೊ ಸೈಂಟಿಸ್ಟ್ ವಾಟ್ ದೇ ವಿಲ್ ಡೂ ವೈಲ್ ದೇ ಆರ್ ಡೂಯಿಂಗ್ ಸಮ್ ರಿಸರ್ಚ್, ಇನ್ ದಟ್ ಟೈಮ್ ದೇ ವಿಲ್ ಹ್ಯಾವ್ ದ ಮಾಸ್ಟರ್ ಪ್ಲೇಟ್. ಓಕೆ, ಒಂದು ಮಾಸ್ಟರ್ ಪ್ಲೇಟ್ ಇರುತ್ತೆ. ಸೊ ಅದ್ರ ತರನೇ ಸೆವೆರಲ್ ಅದರ್ ಪ್ಲೇಟ್ಸ್ ನ, ಆಸ್ ಯು ಕ್ಯಾನ್ ಸಿ ಇನ್ ದ ಡಯಾಗ್ರಾಮ್ ಲೈಕ್ ದಿಸ್, ಅದೇ ತರನೇ ಸೆವೆರಲ್ ಅದರ್ ಡಿಫ್ರೆಂಟ್ ಪ್ಲೇಟ್ಸ್ ನ ಇನ್ಕ್ಯುಬೇಟ್ ಮಾಡ್ತಾರೆ ವಿತ್ ದ ಡಿಫ್ರೆಂಟ್ ಗ್ರೋತ್ ಮೀಡಿಯಾ. ಲೈಕ್ ಇನ್ ಸಮ್ ಗ್ರೋತ್ ಮೀಡಿಯಾಸ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ವಿಲ್ ಬಿ ದೇರ್, and in some growth media nutrients will not be there, or some growth medias might have a antibiotic in it. So ಆ ತರ replica plate ನ the main master plate ಜೊತೆ ಇನ್ಕ್ಯುಬೇಟ್ ಮಾಡ್ತಾರೆ, ಸೊ ದಟ್ ದೇ ಕ್ಯಾನ್ ಫೈಂಡ್ ಔಟ್ ದ ಮ್ಯೂಟೆಂಟ್ಸ್. ಸೊ ಮ್ಯೂಟೆಂಟ್ಸ್ ಯಾವ್ದ್ರಿಂದ ಗ್ರೋತ್ ಆಗುತ್ತೆ ಅಂತಂದೇಳಿ ಕಂಪ್ಯಾರಿಸನ್ ಮಾಡಿ ಫೈಂಡ್ ಔಟ್ ಮಾಡ್ತಾರೆ. ಸೊ ಹಾಗಾಗಿ ಈ ತರ ಡಿಫ್ರೆಂಟ್ ಅದರ್ ರೆಪ್ಲಿಕಾ ಪ್ಲೇಟ್ ನ ಯೂಸ್ ಮಾಡಿದ್ರೆ, ಮಾಸ್ಟರ್ ಪ್ಲೇಟ್ ನ ಇಟ್ಕೊಂಡು ಅನದರ್ ಪ್ಲೇಟ್ಸ್ ನ ಏನ್ ಪ್ರಿಪೇರ್ ಮಾಡ್ತಾರಲ್ಲ ಅದನ್ನ ನಾವು ರೆಪ್ಲಿಕಾ ಪ್ಲೇಟಿಂಗ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಈ ರೆಪ್ಲಿಕಾ ಪ್ಲೇಟಿಂಗ್ ನ ಏನಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತಾರೆ? ಟು ಫೈಂಡ್ ಔಟ್ ದ ಮ್ಯೂಟೆಂಟ್ಸ್. ಓಕೆ, ಮ್ಯೂಟೆಂಟ್ ಕಾಲೋನೀಸ್ ನ ಫೈಂಡ್ ಔಟ್ ಮಾಡಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ತಾರೆ, ಮ್ಯೂಟೆಂಟ್ ಕಾಲೋನಿ. ಮೂವಿಂಗ್ ಆನ್ ಟು ದ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್, 22nd ಕ್ವಶನ್. The female sex organ of patrocospermum is called as option A oogonium, option A oogonium, option B carpagonium, option C female conceptacle, option D female cryptoblast. Let's solve this. We will know what is this patrocospermum. Patrocospermum, it is a red algae which is like purple to red in color. ಸೊ ಅದ್ರದ್ದು ಫೀಮೇಲ್ ಸೆಕ್ಸ್ ಆರ್ಗನ್ ಯಾವುದು ಅಂತ ಕೇಳಿರೋದು. ಫಸ್ಟ್ ಒನ್ ಓಗೋನಿಯಂ. ಓಗೋನಿಯಂ is also a reproductive female sex organ, but this oogonium usually present in the algae and fungi. And coming to the carpagonium is also female sex organ of in the red algae. ಓಕೆ, ಸೊ ಹಾಗಾಗಿ ಆಪ್ಷನ್ ಬಿ ನೇ ಕರೆಕ್ಟ್ ಆನ್ಸರ್. ಬಿಕಾಸ್ ಪ್ಯಾಟ್ರಕೋಸ್ಪರ್ಮಮ್ ಇಟ್ ಈಸ್ ಅ ರೆಡ್ ಆಲ್ಗೆ. ದ ಫೀಮೇಲ್ ಸೆಕ್ಸ್ ಆರ್ಗನ್ ಇವಾಗ ಯಾವ್ದಾಗ್ತದೆ? ಕಾರ್ಪೋಗೋನಿಯಂ ಆಗುತ್ತೆ. ಸೊ ಕಾರ್ಪೋಗೋನಿಯಂ is the female sex organ in the patrocospermum, or you can say in the red algae. And coming to the options is female conceptacle. Female conceptacle, so, the female conceptacle, it is also a specialized cavity present in the marine and freshwater algae that contains the reproductive organ. Coming to the option D, female cryptoblast. The female cryptoblast, it is nothing but it is a sterile conceptacle. Okay, so option B is the correct answer. Moving on to the next question. During the dark reaction of photosynthesis is cyanobacteria. Option A, water is split. Option B, CO2 is reduced to organic compound. Option C, chlorophyll is activated. Option D, 6 C sugars broken into 3 C sugars. ಸೊ ಇಲ್ಲಿ ಫೋಟೋಸಿಂಥಸಿಸ್ ಬಗ್ಗೆ ಕೇಳಿದ್ದಾರೆ. ಐ ಹ್ಯಾವ್ ಡನ್ ಸೋ ಮೆನಿ ವಿಡಿಯೋಸ್ ಆನ್ ಫೋಟೋಸಿಂಥಸಿಸ್. ಇಫ್ ಯು ಆರ್ ಇಂಟ್ರೆಸ್ಟೆಡ್ ಯು ಕ್ಯಾನ್ ವಾಚ್ ಇಟ್. ಸೊ ದ್ಯಾಟ್ ಕುಡ್ ಹೆಲ್ಪ್ ಯು ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದಿಸ್ ಕಾನ್ಸೆಪ್ಟ್. And for the time being, I'm going to explain this. Photosynthesis, it is nothing but it has a two phases. Okay, it has a light phase and it has a dark phase. And in case of light phase, and again this light phase consists of two processes. In this light phase, there is a production of NADPH, means there is a transfer of electrons from water to NADP plus. There is a transfer of electrons from water to NADP plus, and that will converted into NADPH. Okay, that is the first process. And later, second one, there is a conversion of or you can say there is a conversion of radiant energy into a chemical energy, that is called as photophosphorylation, means there is a synthesis of ATP. Okay, those are the light reactions. Coming to the dark reaction, in case of dark reaction, what happens? It is going to utilize the products of the light reaction. That is, it is going to produce the carbon dioxide. So it is going to utilize the light reaction products, that is ATP and NADPH, to produce carbohydrates. So ಹಾಗಾಗಿ the correct answer, so dark reaction ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂತ ಕೇಳಿದ್ರು. So correct answer for this is CO2 is reduced to organic compound. So correct answer for this is option B. CO2 is reduced to organic compounds. Moving on to the next question. The net gain of ATP molecules in glycolysis. Option A is 0, option B 2, option C 4, and option D is 8. So total agi net gain of ಎಷ್ಟು ATPs produced advu in a glycolysis anta kelta irodu. As you can see in the diagram here which is I'm displaying, so this is the chart of the glycolysis alli enagutte first. So there are ಎಷ್ಟು 4 ATPs produce ಆಗುತ್ತೆ. ಟೋಟಲ್ ಆಗಿ 4 ATPs produce ಆಗುತ್ತೆ. ಎಲ್ಲಿ ಅಂದ್ರೆ one is produced at converting the triose bisphosphate into a triose phosphate. Okay, there ATP is formed. Okay, ಅಲ್ಲಿ 2 ATPs formation ಆಗುತ್ತೆ. And then in converting the phosphoenol pyruvate into a pyruvic acid, there also 2 ATPs are going to produce. 2 ಎಷ್ಟಾಗುತ್ತೆ? 4 ATPs are produced. And in 4 ATPs, again here converting the glucose to glucose 6-phosphate, the 1 ATP is utilized. Okay, and again converting the fructose 6-phosphate into fructose 1,6-bisphosphate, there also another ATP is utilized. So 4 ಅಲ್ಲಿ ಕೊನೆಗೆ ನೆಟ್ಟಲ್ಲಿ ಉಳಿಯೋದು ಎಷ್ಟು? 2 ATPs. Okay, 2 ATPs are going to remain. So correct answer for this is option B, that is 2 ATPs. Moving on to the next question. The etiological agent responsible for ticker disease of the groundnut is option A Sarcoraspora species, option A Sarcoraspora species, option B Alternaria, option C Xanthomonas species, option D Puccinia species. Ticker disease of the groundnut ಅಂತಂದ್ರೆ ಶೇಂಗಾ ಲೀವ್ಸ್ ಅಲ್ಲಿ ಈ ತರ ಬ್ಲಾಕ್ ಬ್ರೌನ್ ಕಲರ್ ಸ್ಪಾಟ್ಸ್ ಆಗಿದ್ರೆ ಅದನ್ನ ಟಿಕ್ಕರ್ ಡಿಸೀಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಓಕೆ, ದಿಸ್ ಟಿಕ್ಕರ್ ಡಿಸೀಸ್ ಕಾಸ್ ಆಗೋದು ಬೈ ಸರ್ಕೋರೋಸ್ಪೋರಾ ಸ್ಪೀಸಿಸ್ ಇಂದ. ಸರ್ಕೋರೋಸ್ಪೋರಾ ಸ್ಪೀಸಿಸ್ ಅಲ್ಲಿ 2 ಟೈಪ್ಸ್ ಇದೆ, ದಟ್ ಈಸ್ ಸರ್ಕೋರೋಸ್ಪೋರಾ ಅರಾಕಿಡಿಕೋಲಾ ಅಂಡ್ ಅನದರ್ ಒನ್ ಈಸ್ ಸರ್ಕೋರೋಸ್ಪೋರಾ ಪರ್ಸೊನೇಟಾ. ಸೊ ದೀಸ್ 2 ಸ್ಪೀಸಿಸ್ ಇಂದ ಟಿಕ್ಕರ್ ಡಿಸೀಸ್ ಫಾರ್ಮೇಶನ್ ಆಗುತ್ತೆ. ಟಿಕ್ಕರ್ ಡಿಸೀಸ್ ಗೆ ಅನದರ್ ನೇಮ್ ಇದೆ, ದಟ್ ಈಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಆಸ್ ಲೀಫ್ ಸ್ಪಾಟ್ ಡಿಸೀಸ್ ಅಂತ ಹೇಳ್ಬೋದು. ಸೊ ಹಾಗಾಗಿ ದ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಫಾರ್ ದಿಸ್ ಇಸ್ ಆಪ್ಷನ್ ಎ, ದಟ್ ಈಸ್ ಸರ್ಕೋರೋಸ್ಪೋರಾ ಸ್ಪೀಸಿಸ್. ದ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಅಪಿಯರ್ ಆಸ್ beaded structures at option A leptotin, option B pagetin, option C diakinesis, option D telophase. If you think this is chromosome ಅಂತ ಅನ್ಕೊಂಡ್ರೆ, ಸೊ ಈ ತರ ಬೀಡ್ಸ್ ಇರುತ್ತೆ, ಈ ತರ ಓಕೆ, ಬೀಡೆಡ್ ಲೈಕ್ ದಿಸ್ ಬೀಡ್ಸ್ ಇರುತ್ತೆ. ಈ ತರ ಬೀಡೆಡ್ ಆಗಿ ಕಾಣೋದು ಯಾವಾಗ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಸೊ ಲೆಪ್ಟೋಟಿನ್ ಅಂತಂದ್ರೆ ಲೈಕ್ ಇಟ್ ಕಮ್ಸ್ ಇನ್ ದಿ ಮಿಯೋಸಿಸ್ ಫಸ್ಟ್. ಓಕೆ, ಪ್ಯಾಚೆಟಿನ್ ಈಸ್ ಆಲ್ಸೋ ಕಮ್ಸ್ ಇನ್ ದಿ ಮಿಯೋಸಿಸ್ ಫಸ್ಟ್ ಅಂಡ್ ಡಯಾಕಾನಿಸಿಸ್ ಆಸ್ ವೆಲ್, ಅಂಡ್ ಟೀಲೋಫೇಸ್ ಫಸ್ಟ್. ಆಬ್ವಿಯಸ್ಲಿ ಇದು ಮಿಯೋಸಿಸ್ ಫಸ್ಟ್ ಅಲ್ಲೇ ಬರೋದು. ಓಕೆ, ಸೊ ನಮ್ಗೆ 2 ಟೈಪ್ಸ್ ಇದೆ ಅಲ್ವಾ ಸೆಲ್ ಡಿವಿಷನ್, ದಟ್ ಈಸ್ ಮೈಟೋಸಿಸ್ ಅಂಡ್ ಮಿಯೋಸಿಸ್. ಮಿಯೋಸಿಸ್ ಅಲ್ಲಿ ಅಗೇನ್ ಮಿಯೋಸಿಸ್ 1 and ಮಿಯೋಸಿಸ್ 2, and again in meiosis 1 we have again subdivisions like leptotin, zygotin, pagetin, diakinesis. Okay, and then after we are going to get the ಮೆಟಾಫೇಸ್, ಅನಾಫೇಸ್ ಅಂಡ್ ಟೀಲೋಫೇಸ್, ರೈಟ್. ದಟ್ ಈಸ್ ಮಿಯೋಸಿಸ್ 1 ಅಲ್ಲಿ ಪ್ರೊಫೇಸ್ ಫಸ್ಟ್ ಅಲ್ಲಿ ಈ ಡಿಫ್ರೆಂಟ್ ಟೈಪ್ಸ್ ಬರುತ್ತೆ, ದಟ್ ಈಸ್ ಲೆಪ್ಟೋಟಿನ್, ಪ್ಯಾಚೆಟಿನ್, ಡಯಾಕಾನಿಸಿಸ್ ಆ ಥರ. ಆ್ಯಂಡ್ ದೆನ್ ಅಗೇನ್ ಮೆಟಾಫೇಸ್ ಅಂಡ್ ಅನಾಫೇಸ್ ಅಂಡ್ ಟೀಲೋಫೇಸ್. ಓಕೆ, ಬಟ್ ಮಿಯೋಸಿಸ್ ಅಲ್ಲಿ ಡೈರೆಕ್ಟ್ ಆಗಿ ಮಿಯೋಸಿಸ್ 2 ಅಲ್ಲಿ ಪ್ರೊಫೇಸ್ 2, ಮೆಟಾಫೇಸ್ 2 ಅಂಡ್ ದೆನ್ ಅನಾಫೇಸ್ 2 ಅಂಡ್ ಟೀಲೋಫೇಸ್ 2 ಬರುತ್ತೆ. ಸೊ ಲೆಪ್ಟೋಟಿನ್ ಅಲ್ಲಿ ಸೊ ಆಪ್ಷನ್ ಎ ಇಸ್ ದ ಕರೆಕ್ಟ್ ಒನ್. ಸೊ ಪ್ರೊಫೇಸ್ first of the meiosis first, and in that prophase first, in leptotin phase, in that time the chromosomes are going to appear as a beaded structures. Okay, option A is the correct answer. Moving on to the next question. Which of the following bacteria converts nitrites into nitrates? Option A Nitromonas, option B Chromatium, option C Nitrobacter, option D Chlorobium. So the correct answer for this is Nitrobacter. This is the one which is going to convert the nitrites into nitrates. Moving on to the 28th question. The plant cell differ from the animal cell in the absence of option A endoplasmic reticulum, option B ribosomes, option C mitochondria, option D centriole. The correct answer for this is the plant cell is going to differ from animal cell because the plant cell doesn't have a centrioles. Animal cell have a centrioles. Okay, so option D is the correct answer. According to Baltimore system of classification, positive and single stranded RNA virus belongs to which of the following groups? Option A group 4, option B group 3, option C group 2 and option D group 1. To understand this, we should know Baltimore system of classification. Means Baltimore is the scientist who given the Baltimore system of classification of the virus. Okay, is used to classify the viruses based on the type, based on their manner of synthesizing the mRNA. As you can see in the diagram, so ಕ್ವಶನ್ ಅಲ್ಲಿ ಕೇಳಿರೋದು ಸಿಂಗಲ್ ಸ್ಟ್ರಾಂಡ್ RNA ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಸೊ ಫಸ್ಟ್ there are 7 groups in that, in this Baltimore system of classification of the viruses. ಇದರಲ್ಲಿ 7 classes ಇದೆ. 7 classes ಅಲ್ಲಿ ಫಸ್ಟ್ ಒನ್ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNA, ಸೆಕೆಂಡ್ ಕ್ಲಾಸ್ ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ DNA, and ಥರ್ಡ್ ಕ್ಲಾಸ್ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ RNA, ಫೋರ್ತ್ ಕ್ಲಾಸ್ ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ RNA, and fifth one single stranded RNA, and sixth one single stranded RNA positive, seventh one double stranded DNA. Okay, which has positive and negative. So idike correct answer banbitu fourth one, you can see the single stranded RNA. So it comes in the fourth classification. ಇದನ್ನ ನಾನು another video ನಲ್ಲಿ in detail ಆಗಿ solve ಮಾಡಿದೀನಿ. ನಿಮಗೆ more information ತಿಳ್ಕೋಬೇಕು ಅಂತ ಇತ್ತು ಅಂದ್ರೆ you can go and watch that video. I have solved this question. I had made this video in detail. Option A is the correct answer, which class single stranded RNA virus ಯಾವ್ದ್ರಲ್ಲಿದೆ group 4 ಅಲ್ಲಿ ಇದೆ. Moving on to the next question, 30th one. Bacterial blight of paddy is caused by option A Xanthomonas oryzae, option B Xanthomonas campestris, option C Xanthomonas malvacearum, option D Erwinia amelovera. ಇಲ್ಲಿ ನೋಡಬಹುದು Xanthomonas paddy ನಲ್ಲಿ ಸೊ ಈ ತರ white patches, you can see the white patches. So this bacterial blight of the paddy is caused by Xanthomonas oryzae. ಸೊ ಈ ತರ ಸ್ಪಾಟ್ಸ್ ಕಾಣ್ತಾ ಇರುತ್ತೆ. ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಈ ಡಯಾಗ್ರಾಮ್ ಅಲ್ಲಿ ಮೇ ಬಿ ಇನ್ ಡೀಟೇಲ್ ಯು ಕ್ಯಾನ್ ಸಿ ಈ ತರ ಒಂಥರ ಡ್ರೈ ಆಗ್ಬಿಟ್ಟಿರುತ್ತೆ. ಸೊ ಇಟ್ ಈಸ್ ಕಾಸ್ಡ್ ಬೈ xanthomonas veraceae. Option A is the correct answer. Okay, that is xanthomonas veraceae. Moving on to the next question. The etiology agent of head smut of sorghum is option A Cercospora arachidicola, option B Pyracularia griseae, and option C Sclerospora graminicola, option D Spacillotheca sorghum. ದ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಹೆಡ್ ಸ್ಮಾಟ್ ಆಫ್ ಸೋರ್ಗಮ್ ಅಂತಂದ್ರೆ ಏನು ಅಂತ ನೋಡೋಣ. ಸೊ ಇಲ್ಲಿ ನೋಡ್ಬೋದು, ಕನ್ನಡದಲ್ಲಿ ಏನಂತ ಹೇಳ್ತೀವಿ, ಜೋಳ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಜೋಳ ಅಂದ್ರೆ ನಮ್ಮ ಎಕ್ಸ್ಪೆಷಲಿ ನಾರ್ತ್ ಕರ್ನಾಟಕಕ್ಕೆ ತುಂಬಾನೇ ಇಷ್ಟವಾದ ಫುಡ್. ರೊಟ್ಟಿ ಇಲ್ಲದೆ ಜೀವನನೇ ನಡೆಯಲ್ಲ ಅಂತ ಅನ್ಸುತ್ತೆ, ಬಿಕಾಸ್ ವಿ ಲವ್ ರೊಟ್ಟಿ ಅಷ್ಟೊಂದು ಇಷ್ಟ ನಮ್ಗೆ ಎಕ್ಸ್ಪೆಷಲಿ. ಸೊ ಈ ಜೋಳಕ್ಕೆ ಬರ್ತಕ್ಕಂಥ ಒಂದು ಡಿಸೀಸ್ಗೆ ನಾವು ಹೆಡ್ ಸ್ಮಾಟ್ ಆಫ್ ಸೋರ್ಗಮ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಇಲ್ಲಿ ನೀವು ನೋಡ್ಬೋದು, ದಿಸ್ ಇಸ್ ದ ಒನ್ ವೈಟ್ ಕಲರ್ ಅಲ್ಲಿ ಇದೆ ಸೋರ್ಗಮ್. ಇದು ನಾರ್ಮಲ್ ಆಗಿದೆ, ಬಟ್ ಈ ಹೆಡ್ ಅಂದ್ರೆ ಆ ಪೋರ್ಷನ್ಗೆ ಹೆಡ್ ಅಂತ ರೆಫರ್ ಮಾಡಿರೋದು. ಇಲ್ಲಿ ಏನಾಗಿದೆ, ಇದು ಅಫೆಕ್ಟೆಡ್ ಆಗಿದೆ. ಸೊ ಹಾಗಾಗಿ ಬೆಳೆದಿರ್ತಕ್ಕಂಥ ಕ್ರಾಪ್ ಎಲ್ಲ ಏನಾಗಿದೆ, ವೇಸ್ಟ್ ಆಗಿದೆ. ಸೊ ಸ್ಮಟ್ ಫಾರ್ಮೇಶನ್ ಆಗಿದೆ. ಸ್ಮಟ್ ಅಂದ್ರೆ ಇಟ್ಸ್ ಲೈಕ್ ಡಾರ್ಕ್ ಕಲರ್ ಪೌಡರ್. ಯಾವ ಸ್ಪೀಸೀಸ್ ಇಂದ ಬರ್ತಾ ಇರೋದು ಅಂತ ಕೇಳ್ತಾ ಇರೋದು. ಸೊ ಈ ಡಿಸೀಸ್ಗೆ ನಾವು ಹೆಡ್ ಸ್ಮಾಟ್ ಆಫ್ ಸೋಗಮ್ ಅಂತ ಹೇಳೋದು. ಸೊ ಆಪ್ಷನ್ಸ್ ಅಲ್ಲಿ ನೋಡೋಣ. ಸೊ ಇದಕ್ಕೆ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಬಂದ್ಬಿಟ್ಟು ದಿಸ್ ಈಸ್ ಕಾಸ್ಡ್ ಬೈ ಆಪ್ಷನ್ ಡಿ, ದಟ್ ಈಸ್ Spacillotheca sorghum. Okay, ಇದರಿಂದ ಕಾಸ್ ಆಗುತ್ತೆ. ಮೂವಿಂಗ್ ಆನ್ ಟು ದಿ 32nd ಕ್ವಶನ್. An antigen is option A highly specific protein produced by the body in response to a foreign body. Option B a chemical that induces the growth of microorganisms. Option C an antibody produced by the body that stimulates the production of antibodies by the body's immune system. Option D a chemical or a biological substance that stimulates the production of antibodies by the body's immune system. Antigen ಅಂದ್ರೆ ಏನು ಅಂತಂದೇಳಿ ಕೇಳಿದರು. ಇಮ್ಯುನಾಲಜಿ ಬಗ್ಗೆ ನಾನು ತುಂಬಾ ವಿಡಿಯೋಸ್ ಮಾಡಿದ್ದೀನಿ, ನಿಮ್ಗೆ ಇಂಟ್ರೆಸ್ಟ್ ಇದ್ರೆ ನೋಡ್ಬೋದು. ಸೊ ನಾವು ಈ ಕ್ವಶನ್ ನ ಸಾಲ್ವ್ ಮಾಡೋಣ. ಆಂಟಿಜನ್ ಸಿಂಪಲ್ ಕ್ವಶನ್ ಆಗಿದೆ, ಸಿಂಪಲ್ ಕ್ವಶನ್ ಇದೆ. ಸೊ ನಥಿಂಗ್ ಟು ವರಿ ಅಬೌಟ್ ದಿಸ್. ಇದೇನು ಡೀಪ್ ಡೆಪ್ತ್ ಕಾನ್ಸೆಪ್ಟ್ ನ ಏನು ಕೇಳಿಲ್ಲ. ಆಂಟಿಜನ್ ಅಂದ್ರೆ ಏನು? Antigen is one stimulates the production of antibodies. Okay, antigen is a foreign particle which enters our body. Okay, so ಅದು ಬಂದಾಗ ಏನಾಗುತ್ತೆ, ಆಂಟಿಬಾಡೀಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ ಟು ಪ್ರೊಟೆಕ್ಟ್ ಅವರ್ ಸೆಲ್ಫ್. ಓಕೆ, ಸೊ ಅಂಥವ್ಗೆ ನಾವು ಆಂಟಿಜನ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಒಂದೊಂದ್ಸರಿ ಓದ್ಕೋಬೇಕಾದ್ರೆ ಕನ್ಫ್ಯೂಷನ್ ಆಗ್ಬಿಟ್ಟು ದೇರ್ ವಿಲ್ ಬಿ ಕನ್ಫ್ಯೂಷನ್ ಬಿಟ್ವೀನ್ ಆಪ್ಷನ್ ಎ ಅಂಡ್ ಆಪ್ಷನ್ ಡಿ, ಬಿಕಾಸ್ ಅದು ಡೆಫಿನೇಷನ್ ನಾವು ಕನ್ಫ್ಯೂಷನ್ ಅಲ್ಲಿ ಹಾಕ್ಬಿಡ್ತೀವಿ. ಸಡನ್ ಆಗಿ ಓದಿದ ತಕ್ಷಣ ಆಪ್ಷನ್ ಎ ಕರೆಕ್ಟ್ ಅನ್ಸುತ್ತೆ, ಬಿಕಾಸ್ ದಿಸ್ ಇಸ್ ದ ಡೆಫಿನೇಷನ್ ಆಫ್ ದ ಆಂಟಿಬಾಡಿ, ಎ ಹೈಲಿ ಸ್ಪೆಸಿಫಿಕ್ ಪ್ರೋಟೀನ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಬೈ ದ ಬಾಡಿ ಇನ್ ರೆಸ್ಪಾನ್ಸ್ ಟು ಎ ಫಾರಿನ್ ಬಾಡಿ. ಓಕೆ, ಈ ಫಾರಿನ್ ಬಾಡಿನೇ ಆಂಟಿಜನ್ ಅಂತ ಅನ್ನೋದು. ಓಕೆ, ಈ ಆಂಟಿಜನ್ ಫಾರಿನ್ ಬಾಡಿ ರೆಸ್ಪಾನ್ಸ್ಗೆ ಪ್ರೊಡ್ಯೂಸ್ ಆಗೋದು ಆಂಟಿಬಾಡಿ. ಸೊ ಎ ಡೆಫಿನೇಷನ್ ಏನಾಗುತ್ತೆ, ಅದು ಆಂಟಿಬಾಡಿ ಡೆಫಿನೇಷನ್. ಓಕೆ, ಸೊ ಹಾಗಾಗಿ ಆಪ್ಷನ್ ಎ ಅಲ್ಲ. ಸೊ ಇದು ಕೂಡ ಅಲ್ಲ. ಕೆಮಿಕಲ್ ದಟ್ inhibits the growth of microorganisms. No, that is not the correct answer. Antibody produced by the body. Antigen ಅಂದ್ರೆ ಆಂಟಿಬಾಡಿನಾ? ಅಲ್ಲ ಅಲ್ವಾ? That is not the correct answer. So the correct answer ಬಂದ್ಬಿಟ್ಟು ಇದಕ್ಕೆ ಆಪ್ಷನ್ ಡಿ is the correct answer. Antigen is a chemical or a biological substance that stimulate the production of antibodies by the body's immune system to fight against those foreign bodies or antigens. Okay, option D is the correct answer. Okay guys, that's for this video. In the next video I'm going to solve the other questions. I'm hoping this video is going to help you all, and if you like this video, if you learn something from this video, please do share and subscribe. Thank you everyone.